let's go back to Cytoscape. And let's open some of the results here. So you still have the network saved from the other day. And if you did not change any feature finding settings and rerun your feature finding in job, then you should be able to like just proceed with that old network, right? So let's see. I don't know, open recent. Okay, let's see the combined network here. So again, this is where I left off the other day, and very importantly, this network is already combined between like my series, or what was it, Canopus results, you know, and the GMPS output. So I have both, hopefully, molecular formulas as well as like my libraries IDs in there. I kind of made like all like this subset examples. I'm gonna destroy them now and clean this a little bit up here, so it's more, I don't know, nicer to look at. So yeah, hopefully you have something like that. Whether you imported the series results already or not is not so important. I think the main thing I would rely on is like the library IDs anyway. So I imported this as this TSV table that you got as a summary output file. If you want, I can quickly show it again. So let's see. Here we had our lab 15 series output files, right? And there were this Canopus compound summary TSV. So you should have gotten, if you exported the summary files from series, you should have gotten like, I think, 3 files, one for series, one for Canopus, and one for CSI finger ID. And then if you open those in Excel by dragging and dropping in the TSV, you should get like this different columns, you know, with like all like this different information about the classes, molecular formulas, and so on. And then very important, like this first column here has like this ID, you know, column that has like this long strings. And Kai splitted that already for me. By default, Sirius does not split that for you, so you need to do that yourself. So what I did here is I copied that and pasted it in, you know, at the end. And then with like the data tab, I convert text to columns, and I use underscore as a delimiter. You know, and well, now I can delete here everything but the last one, because this would be the feature ID, right? Feature ID. And now, yeah, you can see there was already a feature name. So this was what Kai created. But yeah, I just redid it from that ID string. And now, you know, I could just save this as, I don't know, a modified TSV file. And then in Cytoscape, I go to File, Import Table from File, and then navigate to these results. Output. And then do you see that there's this key as a symbol for like the different columns in this preview? So I need to set this key now to the column that contains the feature ID, right? And again, this is this number that is also in the very first column in your feature table. So this is simply like the index of a given feature. And for us, this is the key to bring like the data together here. And then, yeah, I just hit OK, and then it imports it. Because these tables become like very complicated, you know, because there's so many different columns. What I did also the other day, I actually concatenated a new header name where I put in a prefix Canopus to each of the columns that came from Canopus. I would suggest that you do something like that, especially, you know, so now again, you know, you have now all the possibility of like importing new information, but then it's also your responsibility to like do that in a way that first of all yourself will not get confused later on. So structuring this a little bit in that way with header prefixes, I think, is a good way. So yeah, in my case, yeah, I used Canopus as a prefix, and that way also like now in this big table, all information from Canopus is kind of like brought together. Say what? Yeah, can you read?
Compound summary of formulas.
Okay, yeah, my apologies for the serious people for not naming this very intuitively. So compounds would be CSI figure ID, I think. And then formula summary, that would be series output. I typically would not use the adducts. And then there's Canopus, okay, that has to be, that's like yeah. And then yeah, for each of them, you know, you would need to find this ID column and then split like that string in the individual component. The last one, that is. So once you fix that, so once this is an individual column, then you go to File, Import, and Table from File, right? And then you just select here, I don't know, my modified one here. And then you need to set the key to the column that has this information. So the other day I moved that also to the first one because it automatically then sets it already. So I did not have to choose it. Now in my case, because it's at the end, you know, I would just click on it and then select the key, and then it displays the key here. Not anymore to the first one. So it imported. Also, like I can like change the data type, you know, where the something is integer or string. You know, I can in case it did not recognize this correctly, I can tell it what type of data it is. Normally by default, it does a good job, but we had it in the past that especially between like integer and float, there was some confusion, and you can select this here, you know. Okay. Frank?
Yeah. So if our Canopus was now finished successfully,
yeah. So if you're actually just for a test, you know, if Sirius, like, I don't know, stopped in the middle or so and only processed half of it, you can write the summaries anyway, and then it will just like write it to like the feature where it got to, right? Obviously now, if this is an important data set, you want to make sure that all the features went through. But now for like, you know, test purposes, feel free to do it also with like the partial thing. Okay, and now like that way we imported now this Canopus information or Sirius, you know, we could also import any other information. For example, you know, like the significance scores from your statistical analysis yesterday. So now this would be not an ID, but actually some, you know, sort of like prioritization for the compounds that could be interesting. Okay, now I just need to find the folder here. I think it was somewhere on stats, nice. And then, you know, here there was this output tables. And now I just opened this in here Excel to check out what it is, you know, and now very nice, you know, here, this would be, again, my feature ID. So Oh no, wait. This one here it is. This would be just an index. So actually I don't need that. So I'm just gonna delete that and move that stats ID to the front. So remember they concatenated like the feature number, the mass, and the retention time in this longer string here. So if there would not be a column like this, the same way as with like series, I could also split that other one.
Yeah, can you quickly show again how to split that?
Yeah. So I just click on it and then go to data and then go to text to columns and then select underscore as a delimiter. And then, you know, it just splits it. That's very convenient with Excel. All right. And now, just as a sanity check, because I'm not 100% sure if they use the same job that I loaded into Cytoscape, you know, I can like quickly look at some of the features and see if the mass fits. So here we have 3,488. Feature number has a mass of 151. If I go to my network, go here to like the filter tab, and then, you know, select, what was it, name or so. Oh. Name, and then search for this one. Yeah, perfect. See, it's 151 as a mass here. Let's actually see what yeah, 1118. Oh, 1112. What is that? But the retention time 96841 968. It's maybe did some weird rounding here. Anyway, so now because I have this table already formatted in a nice way, you know, I could just now, well, save this first. And then go to Cytoscape, file, import, table from file, and then now browse to the folder where Philip left this for us. And then again, now because it was in the first column, it set the key automatically there. And I see, okay, the other ones are there. And then I can import it. And now, you know, with the style, I could highlight now features based on whether they were significant. I can also sort, you know, and actually go, I don't know, like the way down from like the strongest changes, and like explore that way my data in a more, yeah, a little bit like systematic fashion. Okay, like yeah, if you want, you can do that while I'm doing it. And for this lab, let's actually sort this here. And then what was the highest number that was the strongest upregulated or produced compound in the algae, right? And then if it was negative, then it was lower. So that was perhaps a suppressed feature in our DOM background. Okay. So now, yeah, I sorted this here simply by clicking at the header. And now I just go all the way till I get to like the library names. So that should be compound name also. Oh boy. Compound name, okay. And now I can simply scroll till I find something. Okay, so that's our first hit. and it is undegaethyl ethylene glycol. Okay, what do you guys think? What could that be? No, that's PEG, polyethylene glycol. And I think that's, well, it is an environmental contaminant we frequently observe, but it's also a very frequent contaminant we see in all sorts of samples. So it's a lubricant that is often used for filter coating, I don't know, in syringes to like make things go smooth. So it's really like widely used and we see it a lot. So if I would see that, I would always be cautious. Doesn't mean that it's not in your sample, but that's a common contaminant, similar to plasticizers. 3-phenyl phosphate. Okay, maybe that could be a plasticizer. But then we have more PEG. All right, and then we have something here that does not. Okay, what is this? So yeah, really, if I don't know it, like the first thing I do is typically I Google it. Huh. Interesting. So that looks like, yeah, an alkaloid. Has like this weird structure, I don't know. But yeah. It's awesome if I can find any context. So often Wikipedia entries are kind of like quick reads, and if this is a common herbicide or so, then it's likely that you will find something on Wikipedia. Obviously, if I don't find much information here, I can like then in a second step go like to Google Scholar or I don't know, SciFinder and mine more in like the scientific literature. But I think often for compounds, if they're kind of like widely known, you will get good hits in Wikipedia and so on. Then if I don't find anything but have SMILES code, I visualize it in ChemDraw or ChemSketch, what I showed you the other day, which at least gives me like a kind of like idea from a chemical perspective. Let's see if there is SMILES code. No INCHI, so that means probably also no SMILES code. No, but there's another compound a little bit under it where there was SMILES code. Okay, without going to look at the name now, I will go straight to ChemSketch because that's what I installed here. And go to, how was it? Tools, generate structure from SMILES, and then I will paste this in. And oh, what do you think that could be? I think that could be, yeah. That looks like very much like a plasticizer, or like flame retardants have often like this type of mergers. So also not necessarily something I would assume should be made by Synechococcus. All right, so let's go back to the network. And let's go farther down. And see, now we're already like really deep down, right? So this is not, if I would have looked at the top 50 features, I probably would have not seen anything with like a library hit that could be interesting. Okay, and now there is something that, well, I would assume that could be made by Synechococcus. Well, okay, so yeah, that's a fatty acid. Well, and then we detected it as a methyl ester. That is, yeah, could be something natural. And then, okay, some more fatty acids. Okay, I don't know what this is. But yeah.
So there's other top hits, just flame retardants and plasticizers. How did they get in there at the significant level?
That is a very good question. I don't know, but I would assume that like because we bought that standard as kind of like from an algae farm or so as a just like reference material. I don't know if they have like some, I don't know, coatings on their filters for harvesting it, you know, stuff like that. It could have also been obviously in the background in the ocean water, but then it should have not been like such a significant difference. But yeah. Yeah, there's very few typical natural products from Synechococcus annotated. Interesting. But you know, maybe that molecular space is just like not very well described, right? So again, most of the top features, they did not have any ID, right? So, and that is kind of, I think what like doing like non-targeted metabolomics, we have to be comfortable with is that the majority of the compounds we detect do not have MS IDs yet. So now we can of course ask, okay, but maybe they have interesting molecular formulas, you know, or maybe they have interesting compound classes by cannabis. So we can like scroll here, like to like the cannabis part, you know, and then yeah, we get a little bit more information. And then if there's some like terpenoids, you know, things like that, and there's many terpenoids, you know, that kind of like may tells you, well, there's maybe this class of compounds produced. So I don't know if there was any in silico hits in terms of structure. Oh, I did not import that yet. But yeah, like a lot of like, you know, class level annotations. Again, if you describe it, you know, make it clear this is level 3. So we're far away from a ID'd compound here, but you know, we may get some insights. All right. Cool. So yeah, like this table way is one way to look at it. Again, I could also, you know, use the like the shapes of the nodes or colors also to like highlight what is significant here in the network. So when I go to the style tab, and remember, like I put in all these mappers last time. So for example, for size, I can just hit on the trash button and then actually now add a new one, such as stats, significant, or what was it? Let's take the p-value, and then, you know, like, okay, the higher the p-value, then it's less significant. Okay, and then, you know, I can go through and then, oh yeah, I see already very nicely that the ones that are big, you know, they have like this clear pattern in the pie chart. So the ones that are small, you know, they're kind of like mixed. So maybe there was not much of a significant difference. And then the ones that are big, normally the biggest piece of the pie is the green one. So now my statistical analysis and this visualization in my kind of eye that really is good in like scanning these patterns quickly work together. And yeah, hopefully there should be not too much like of a disagreement. So now again, with like the shape, you know, I can eventually indicate what was a, you know, analog. So now that use the rectangle. What was a library ID compound, you know, and that way I can explore this also here in the network. I personally prefer the table because this is more systematic, but you know, later for visualization purposes, obviously that is really nice. If you indicate directly abundance in the node of like your different sample groups. Okay, so now, I don't know, I would say take a couple of minutes and try this. I scroll in the meantime a little bit through till I find something exciting. And then we move on with visualizing this down the line. Who of you managed to import this into their network? Why let you explore too much on your own? Who actually has something to explore right now on their computers? Okay, a couple of people. Okay, who's like struggling with something and doesn't know how to proceed from there? Okay, where you stuck? Oh, but they should. So the identifications from GMPS, they should be in there by default if you open the GraphML file. So then they're already in there. Okay. Maybe don't worry about Sirius so much right now, but try to import the stats rather, because I think that right now I want to like look mainly at like ID'd compound by library matches. So those are like the most valuable ones anyway. And then later on you can try with the Sirius TSV. Okay, any other problems? Yeah, just import in the stats file. Because yeah, I think for the exercise later on, this would be something I would also suggest, you know, because that way you actually can like decide on what you think are like the most important features. Yeah. Just click somewhere in the middle where there's no node. So if you just click in the blank space, then it should unselect. Yeah.
Is there a way in Cytoscape to filter the table, for instance, let's say stats is significant and has a compound name, so it makes it easier to
Yeah. Yeah. So that's kind of like what I did here right now is like navigating, right? You can have this filter tab and then, okay, let's do what you just said. I want to now, besides using the filter function right away in the table, I want to actually set some general filters. So I could use here this binary information from the stats table. So I think there was this significant stats significant, right? And then there is yes and no. And let's say we only want to look at yes. And if I hit enter, then it should now only display things that were significant. Oh, actually, it's significant. So let's change this. Right. Oh, it only highlights it. It does not subset in the table. Okay, so then, yeah, what I would do then is you can sort here by significant and non-significant, and then perhaps create a new network just on the significant ones. So that you can do by, selecting everything that is significant here. So you right-click, select nodes from selected rows, go here to network, and then file new network from selected edges, selected nodes. And then you would get a subset of the network. So then we can rename this and say, okay, these are the significant ones. Yeah. One way of doing it. Okay. I think before we move to the lunch break, I just want to show you now what I do if I found something that I thought was like interesting, right? So I don't know. Here, so I created a subset already, but let's assume you're still navigating in your main network, and you now found here this network that has a lot of features that were significant and may also have some interesting IDs. So what I would do is I would mark the full thing by holding Control and then making this box with the mouse around it. And then in the table, you should see already now only like the entries for like those molecules that I have selected. Not, let's, okay, try this again. Yeah. And then I go to File and say New Network from Selected Node, Selected Edges. And then now it makes a new network here where it only displays this one, right? And now, I don't know, depending on like the identity, of them, you know, I could give that a meaningful name. I don't know. Compound name. Oh yeah. So these are like some fatty acids. So I don't know. I'd call it fatty acid network 1. Okay. And now I'm already like a step closer towards like reporting this, because I think now, okay, this is interesting from a, you know, abundance difference. So like particularly these ones here, right? They have very clear patterns with regards that they're like more abundant in the actual algae containing samples. There's a lot of like unknowns too. And then, you know, there's a couple that are like really small because they were not significant, but maybe I'm not like too much interested in them now. So now what I gonna do next with this? So, well, I would like look, how do the features look, right? I would go to the dashboard and like with the masks and like the files that I know where they should be in, I would generate an XIC and kind of like look at them. And then there's actually 2 nodes that kind of like caught my attention in that regard, you know, and that would be here, this 217 ones, because they have very similar masses, right? So now a question that I would wonder is, are those actually really 2 resolved chromatographic peaks, or are they maybe split features, right? Because if they're split features, then, well, maybe I should not report them as like 2 individual nodes, but I don't know, merge them. Or if that happens very often, then maybe I should reconsider my feature finding. So then that would be, of course, bad, because then I have to go back into the entire thing again. Fortunately, all your data analysis was in some sort of like script form. So if you have to redo this, it should be fairly fast, right? To reprocess and then push through like the pipeline. But yeah, so for now, okay, let's assume I go to the dashboard and they're like truly 2 individual peaks, and that's a real thing. And I don't know, maybe I also know that this by chemical context is like a high fatty acid containing Synechococcus culture. And I want to like showcase this now as one of like the results in my paper. Then of course I want to now get this out into a figure, right? And Allegra showed you the, in our opinion, go-to tool, which would be like a vector graphic program to further manipulate that. But before I do that, I would actually try and already like get this ready so that I don't have to do much of like manipulation downstream. Because first of all, I'm lazy, so I don't want to like frickle too much in Illustrator. And then also it's a question of reproducibility, right? The less I have to like change there and the more I have in my original Cytoscape file, the better. Because this file I just post in like to like the repository with all my other files as source data, and then people who just for free download Cytoscape and they can like open this and reproduce it. That way, I think also organizing the subnetworks that were like significant in like new networks here, in like on the left here, you know, that makes it really easy because I can just jump between what is important. But then also things like, you know, changing the size of the labels is much more intuitive here than in Illustrator, because there I would click them and then change like the font size. So here through the style button again, I can like do that like just like very easy. So I would try and find here under node label size. Let's see. Label font size should be here. Here we go. And I just can, you know, increase this to 20. And then, okay, you can see it hopefully already a little better, but, you know, also for people with not as good eyesight, you know, you want to make sure to make this like nice and large. And often, you know, this is kind of a problem with figures that just like the labels are like too small and people have a hard time reading it. If you have too many nodes, you know, then I would rather suggest to not put labels on it before they're so small and nobody can read it, because that's not meaningful. then often what we do is that we put a big network in the SI where we don't have like any size limitations for people who want to know this in detail. But yeah, so then let's say I'm happy with this. Then how do I get it out of here? So I zoom to like a scale that this is fully covered. And then I go to File, Export, Network to Image. And then very nicely, I can either select PNG as a pixel-based image, or SVG, or also JPEG or PostScript, so if you prefer those. But yeah, what we typically use is SVG. And then you specify, you know, like the file name and like the location, and then you hit OK. So now, if I'm not sure if I want to use labels later, I would directly remove the labels and also export it to image, and then just like without labels. Because again, I'm lazy, and like manually selecting all like the labels and deleting them in Illustrator for like a big network actually takes really long. So just in case, I would directly do it. If you do it now from the same view, the files will be identical, and you can like simply like replace them. Okay, and then same, if this would be now for my presentation, you know, where I don't want to manipulate this anymore, then I would already export it as a PNG, and then this goes straight into PowerPoint. So again, therefore, export network to image, and then I would select PNG. Now, the resolution might be important, right? And I can like kind of like change this with this bar. Yeah, this, yeah, I can also provide a resolution. Again, typically for print, you would have 300 dpi. If this is only for an online presentation or so, then 150 dpi are fine. And then obviously like the size of your poster or like screen or whatever. So let's assume this is for a poster, and you know, we want to 20 inches wide. Then I would just say, okay, export. And then hopefully here in the stats folder, there should now be my network as a nice image. And if that were for a presentation, I would give here, the resolution is really nice, so nothing is blurry or pixeled. Again, if I would want to put this in a larger figure into context, you know, I would use the SVG. Oops. And then manipulate this probably with Illustrator or with PowerPoint or Inkscape or whatever you prefer. There's also CorelDRAW. We did not mention that this morning. I know a lot of people use that too. So that would be another vector graphic program. Cool. I guess that's all I wanted to show you with regards to Cytoscape visualization. After the lunch break, Absa is going to show you how to export SVG directly out of like the Jupyter notebooks with ggplot.